Hi everybody. So in the competitive class, let us start the one more chapter, that is microbes in human welfare. Okay, let's go to the question number 1. Which gas is responsible for puffed up of appearance? Okay, puffed up appearance of idli and dosa batter or duff. Okay, obviously it's a very simple and a familiar question. It is due to carbon dioxide. But which organisms are responsible for the production of CO2 in idli and dosa batter? Remember, in idli and dosa batter preparation, we are not going to add any yeast. Okay, don't think it is due to yeast. So yeast is not responsible for the production of that batter, idli and dosa batter. Okay, so the two organisms are responsible for that. It is Streptococcus, Streptococcus species. Okay, so predominantly it was said to be Streptococcus, but a recent investigation showed that instead of Streptococcus, Leuconostoc is abundant. Leuconostoc species is main responsible for that smoothness or puffiness of idli and dosa batter. Okay, that is the Leuconostoc, and secondly it is a Streptococcus species. Streptococcus species. Okay, but in the book and all they mentioned this Streptococcus only. So remember, the Streptococcus is responsible for the production of CO2 and puffed appearance of duff. Okay, COCCU. Okay, so let's move on to the next question. It is not dirt, it is duff. That's question number 2. Which bacterium helps in the production of Swiss cheese? Okay, so Swiss cheese is produced due to of course, if you heard the term Swiss cheese, you can remember the Tom and Jerry cheese. Okay, the Tom and Jerry cheese is nothing but the Swiss cheese. Okay, so there should be a space here. Propionibacterium. Here there is a space. Trichoderma. There is a space. Saccharomyces cerevisiae, Aspergillus. Okay, there is a space should be there. So Swiss cheese, the whole. Of course, in a Swiss cheese we can find that holes like this. That holes are due to the organism. These holes in a Swiss cheese is due to the activity of Propionibacterium shermani. Remember, the Propionibacterium shermani is responsible for that large holes in Swiss cheese. So that produces the large holes due to the carbon dioxide production. Okay. So one more cheese is there called as Roquefort cheese. That Roquefort cheese is ripened with Penicillium roqueforti. Roquefort, the name, genus name and the species name is Roqueforti. So Penicillium roqueforti is responsible for the production of one more cheese, Roquefort cheese. Okay. Then a Trichoderma polysporum, from this one chemical is isolated called cyclosporin A. It can be used in treatment. It can be used in the treatment of persons who have undergone organ transplantation. Saccharomyces cerevisiae, it is the most common yeast called as baker's yeast or fermenting yeast. Then Aspergillus niger, it is a major source of production of citric acid. Okay, the tasting powder, that citric acid is produced from the Aspergillus niger itself. Okay, so Swiss cheese, Propionibacterium shermani. So in this chapter you need to remember so many organism names along with their product and their useful. What are the activities of those organisms? Let's move on to the next question. Question number 3. Yeah. Streptokinase, use it as a clot buster. You need to remember this. Streptokinase, clot buster. So during the patient who undergone heart attack, in the emergency case, the doctor they gave the injection. The first injection they had given is the streptokinase itself, because it removes the clots in the blood vessels. Okay, so blood vessel clot remove ಮಾಡಲಿ blood flow free ಆಗಿ ಮಾಡಲಿಕ್ಕೆ streptokinase is preferred. That streptokinase, you can easily remember the organism name. See the strepto is a genus name. The name is derived from genus name itself. See there, strepto, the same name, strepto. So it is streptococcus. Okay, it's not staphylococcus, then not lactobacillus, not saccharomyces. Okay, so it is streptococcus species. So from streptococcus species you can isolate a streptokinase. It removes the clots, okay, in the blood vessels, act as a clot buster. Okay, so streptokinase as a clot buster. Okay, yeah, next come to the next question. So statins, so one more, not statins, it is statins, S-T-A-T-I-N-S. Statins, a bioactive molecule inhibiting the enzyme responsible for synthesis of Okay, so statins can lower the blood cholesterol level in the person. Okay, so like atorvastatins, usually it is given to the patients who have BP, blood pressure, high blood pressure. Those patients are need to take this statins regularly to lower the cholesterol levels. Okay, so statins reduces the cholesterol level by inhibiting with the enzyme which is synthesize the cholesterol. Okay, so cholesterol synthesis na reduce ಮಾಡುತ್ತೆ statins. Okay, so remember it, important. Okay, next question number 5. Ganga and Yamuna action plan is initiated by, okay, so Minister of Environment and Forest, Minister of Agriculture, Minister of Human Welfare, none of the above. So these are all the not options. Ministry of Environment and Forest, so initiated the Ganga and Yamuna action plan to clean the rivers. Okay, so no Ganga and Yamuna actions to clean the Ganga river as of now, but COVID cleaned it clearly. Okay, so COVID make the Ganga river more cleaner than any action plans. Okay, yeah. So next question 5. Next question 6. Methanogenic bacteria are present in. So methanogenic bacteria, those produces methane, are called as methanogens. So methanogens are archaebacteria. Okay, they are archaebacteria. They have the ability to produce methane gas. They are present in anaerobic sludge or rumen, a part of the stomach of a cattle, both A and B, and none of the above. So anaerobic sludge digester. Okay, so in a effluent treatment plant, in the first tank, the debris were removed. Effluent was sewage water is made into dump on a filters. Then physical debris were removed. The part which is allowed to settle, some part is going to settle at the bottom. This is called a primary sludge. So it is primary sludge. Okay, so then water is pumped into another tank where it is allowed for aeration, called as aeration tank. So in the aeration tank, it is fitted with the impellers or fans for continuous agitation. Then when the BOD is reduced, so here BOD is more here, but BOD when it is reduced, this water is pumped into another tank called as a sediment tank. So in the sediment tank, the flock is going to be settled at the bottom, which consisting of aerobic bacteria there. So this is called a secondary sludge. So this is secondary sludge. So both of primary as well as secondary sludge, they are made mix into one more tank. That tank is called anaerobic digester. In the anaerobic digester, in this anaerobic digester and aerobic digester. In this anaerobic digester, methane gas is going to be produced due to the activity of methanogens. Okay, so this is sewage treatment plant. So whatever the thing it is settled there, which is present on the water surface, is called flock. Okay, so this is one part of it is get pumped back into the tank, that is called activated sludge. So it is activated sludge, which consisting of mainly aerobic bacteria and aerobic bacteria and fungal mat. Okay, so in this tank there is anaerobic digester where methane gas is going to be produced due to the activity of these methanogens. Okay, so it is a simpler way to remember the sewage treatment. So this is the primary treatment, up to here primary treatment, and this is the secondary treatment. Okay, so after purification, water is released into the water bodies. After quaternary treatment also, sometime tertiary and quaternary treatment. So your primary and a secondary sludge is pumped back into one tank is called as anaerobic digester. Okay. Yeah, so it is present in anaerobic digestion or anaerobic sludge also. So at the bottom, finally, after gas is going to be produced, it gets settled at the bottom and it can be taken out. It can be used as manure. It can be used as manure, or it can be used as compost as well. Okay, so after drying it. Okay, so rumen of a cattle. Of course, in a rumen of a cattle also methanogens are present. Okay, so in case of anaerobic sludge also methanogens are present. In rumen also methanogens are present. So they are present on both. So option A and B is the right option for this question. Okay, so that's it. Let's move on to the next question. Question number 7. Okay, so the technology of biogas production was developed in India mainly due to the efforts of, so that biogas plant, which consisting of a cement tank where it is a digester actually. So it is completely covered with one metal tank where it is fitted with the pipe for connection to house. Okay, it is a gas outlet where it is fixed with one tank where slurry is prepared and allow it into dump. Then outlet is there. Okay, so whatever the slurry is dumped here, it is going to be digested here due to the activity of methanogens. Methane is gas is going to be produced. When the gas is produced, there will be a lifting of this metal tank. When gas is not there, it is present at the bottom. Along with when it lift the tank, it overflow into the tank. Okay, overflow tank. So where it is outlet actually. So it was taken out, dried and used as a manure. So this technology of production of this biogas, okay, is called as biogas plant, commonly called as gobar gas. So that plan of this gobar gas was developed by the two agencies. One is Indian Agriculture Research Institute IARI and Khadi and Village Industries Commission called KVIC. The effort of these two agencies are commissioned together responsible for the designing of this biogas plant. Okay, so the right option is this one. So National Botanical Research Institute is a premier research institute for plant science. So plant science research were going on this National Botanical Research Institute. Okay. Then Indian Council of Medical Research ICMR. So premier medical research institute. They have a lot of research institution comes under that Indian Council of Medical Research. Then Indian Council of Agricultural Research. So this ICAR is responsible for monitoring the hybridization or releasing of new varieties. Okay, that is ICAR. Okay. Yeah. Next, go to the next question. Question number 8. BOD refers to bacterial oxygen demand, biochemical oxygen demand, biochemical operation demand, biological organism demand. Okay. So when the water is pumped into the tank after filtration, after removal of physical debris, BOD is high here. BOD is too high. When this water is pumped into aeration tank, okay, so in the aeration tank it allows for aeration continuously. So aeration in the sense oxygen is provided continuously. So after few days the BOD of this water is gradually decreased. Okay, so remember higher amount of BOD is high amount of BOD. BOD is more means more pollution. Okay, less BOD, less pollution. No BOD, pure water. Okay, so water which is pure from any organic matter which have no BOD, biological oxygen demand is very less. Then what is that BOD? Biochemical oxygen demand, or it is also termed as biological oxygen demand by the bacteria. Okay, so biological oxygen organism demand, biological oxygen demand. Okay, so why the bacteria need oxygen? Okay, so here whatever the organic matter, so organic matter, so let me explain what is actually the BOD is. Organic matter which is present in 1 liter of water. Suppose this is a 1 liter of water which contains some organic materials or organic waste. So this organic waste includes, organic matter includes, so excreta of different organisms, agricultural waste, industrial, food industrial waste or household waste. So ಎಲ್ಲದು ಇಲ್ಲಿ dissolve ಆಗಿರುತ್ತೆ. Okay, it rich with organic matter. So this organic matter can be made into breakdown by aerobic bacteria. That aerobic bacteria break down that organic matter and reduces that organic matter amount in the water. ಎಷ್ಟೊಂದು ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ ಇರುತ್ತೆ ಅದನ್ನೆಲ್ಲ ಬ್ರೇಕ್ ಡೌನ್ ಮಾಡಿ ಈ ವಾಟರ್ನ ಕ್ಲೀನ್ ಮಾಡಿಬಿಡುತ್ತೆ. ಫಾರ್ ದಟ್ ಪರ್ಪಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ವಿಚ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಿಸ್ ವಾಟರ್ ದೇ ರಿಕ್ವೈರ್ ಆಕ್ಸಿಜನ್. So they demand oxygen. If we give some amount of oxygen, we can clear the water. They demanding for the oxygen. So that amount of oxygen which is required to clean up this water, water free from organic matter, that demand is nothing but oxygen demand. Okay, so if we give that much of oxygen to the bacteria, they made the water clean. So biological oxygen or biochemical oxygen demand is the amount of oxygen required by the bacteria which are present in the sewage water. Amount of oxygen required by the bacteria to oxidize the organic matter. ಅಲ್ಲಿರೋ ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ನೆಲ್ಲ ಕ್ಲೀನ್ ಮಾಡೋಕ್ಕೆ. The amount of oxygen required by the bacteria to oxidize 1 liter of effluent. 1 liter of effluent is called as biological oxygen demand or biochemical oxygen demand. Okay. So ಕೇಳಿದಷ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕೊಟ್ಟರೆ they can clean up the water. So ಎಷ್ಟು ಕೇಳ್ತಾ ಇದ್ದಾವೋ that is demand. Okay. Yeah, it is biochemical oxygen demand. So remember BOD of drinking water should be 0. ಸೊ ಅದ್ರಲ್ಲಿ ಏನಾದರೂ 0 ಇಲ್ಲ ಇನ್ನೂ ಜಾಸ್ತಿ ಇದೆ ಅಂದರೆ ಸಮ್ ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್, ವೇಸ್ಟ್ ಮೆಟೀರಿಯಲ್ ಇಸ್ ವೇಸ್ಟ್ ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ಪ್ಯೂರ್ ವಾಟರ್ ಅಂತ ಅರ್ಥ. ಓಕೆ, ಯಾ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ನಂಬರ್ 9. ಸೊ IPM is International Pest Management Program, International Pesticide Management Program, Integrated Pest Management Program, Internal Pest Management Program. So the right option is this one, Integrated Pest Management Program. So what is an integrated pest management program? So it is a overall program need for pest management. Okay, so ಈಗ ಪೆಸ್ಟ್ಗಳನ್ನು ಇನ್ಸೆಕ್ಟ್ ಪೆಸ್ಟ್ ಇರ್ಬೋದು ಅಥವಾ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ ಆಫ್ ಪೆಸ್ಟ್ಸ್ ವಿಚ್ ಆರ್ ಅಪಿಯರ್ಡ್ ಇನ್ ದ ಕ್ರಾಪ್ ಪ್ಲಾಂಟ್ಸ್, ಆ ಪೆಸ್ಟ್ಗಳನ್ನೆಲ್ಲ ಕಂಟ್ರೋಲ್ ಮಾಡಲಿಕ್ಕೆ ಇಂಟಿಗ್ರೇಟೆಡ್ ಅಂದರೆ ಆಲ್ ಟುಗೆದರ್ ಎಲ್ಲ ಸೇರಿಸಿ ಏನೇನು ಮಾಡೋಕ್ಕೆ ಸಾಧ್ಯನೋ ಅಷ್ಟನ್ನು ಮಾಡುವಂಥದ್ದು ಇಂಟಿಗ್ರೇಟೆಡ್ ಪೆಸ್ಟ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್. ಸೊ ದಿಸ್ ಇಂಟಿಗ್ರೇಟೆಡ್ ಪೆಸ್ಟ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ದಟ್ ಇನ್ಕ್ಲೂಡ್ಸ್ physical method of control, disease control ಅಥವಾ insect control by physical method, or it includes biological methods. Okay, so ಇಷ್ಟೆಲ್ಲ ಮೆಥಡ್ಸ್ಗಳು ಇರುತ್ತೆ. ಸೊ ಫಿಸಿಕಲ್ ಮೆಥಡಲ್ಲಿ ಲೈಕ್ ರೋಗಿಂಗ್. ಸೊ ರೋಗಿಂಗ್ ಅಂದರೆ ಡಿಸೀಸ್ ಬಂದಿರೋ ಪ್ಲಾಂಟ್ಸನ್ನು ರಿಮೂವ್ ಮಾಡೋದು, ಇನ್ಸೆಕ್ಟ್ ಪೆಸ್ಟ್ ಆಗಿರೋ ಅಟ್ಯಾಕ್ ಆಗಿರುವಂಥ ಪ್ಲಾಂಟ್ಸ್ಗಳನ್ನು ರಿಮೂವ್ ಮಾಡೋದು, ದಟ್ ಈಸ್ ಅ ರೋಗಿಂಗ್. ಆ್ಯಂಡ್ ಟು ಕಂಟ್ರೋಲ್ ದ ಇನ್ಸೆಕ್ಟ್ ಪಾಪ್ಯುಲೇಷನ್ ದೇ ಕ್ಯಾನ್ ಯೂಸ್ ದ ಟ್ರ್ಯಾಪರ್ಸ್, ಇನ್ಸೆಕ್ಟ್ ಟ್ರ್ಯಾಪ್ ಮಾಡಿ ಅದನ್ನು ಕಿಲ್ ಮಾಡುವಂಥದ್ದು. ಸೊ ದೋಸ್ ಆರ್ ಆಲ್ ಕಮ್ಸ್ ಅಂಡರ್ ಫಿಸಿಕಲ್ ಮೆಥಡ್. ದೆನ್ ಬಯಾಲಜಿಕಲ್ ಮೆಥಡ್. ಬಯಾಲಜಿಕಲ್ ಮೆಥಡ್ ಅಂದರೆ ಗ್ರೋಯಿಂಗ್ ರೆಸಿಸ್ಟೆಂಟ್ ವೆರೈಟೀಸ್, ಗ್ರೋಯಿಂಗ್ ರೆಸಿಸ್ಟೆಂಟ್ ವೆರೈಟೀಸ್. ಓಕೆ, ಅಥವಾ ಬೈ ಯೂಸಿಂಗ್ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ನ ಯೂಸ್ ಮಾಡಿ, ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ ಯೂಸ್ ಟು ಕಂಟ್ರೋಲ್ ದ ಡಿಸೀಸ್. ಓಕೆ, ದೆನ್ ಯೂಸಿಂಗ್ ಆಫ್ ಪ್ರಿಡೇಟರ್ಸ್ ಟು ಕಂಟ್ರೋಲ್ the diseases ಅಥವಾ insects. Okay, so all together these methods are called as integrated pest management. So ಇಲ್ಲಿ predators ಗೆ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ಬೋದು. To control aphids we can use ladybird beetle. This ladybird beetle can eat up this aphids. Okay, so ಅದೇ ಥರ mosquito population can come into lowering by using dragonflies, because these dragonflies can eat up the larvae of these mosquitoes. Okay, so they are butterflies can be controlled by using Bacillus thuringiensis. Okay, spores na use ಮಾಡಿ we can kill that. Okay, so these are all the examples for predators. Okay, of course microorganism ಬರುತ್ತೆ ಎಕ್ಸಾಂಪಲ್ ಇದು PT ಯೂಸ್ ಮಾಡುವಂಥದ್ದು. ಸೊ ಲೈಕ್ ದಿಸ್ we can use so many other alternatives, so other than chemical method. So chemical method, so this is the final method they are going to try. ಒಂದು ವೇಳೆ ಇದರಲ್ಲೂ ಕಂಟ್ರೋಲ್ ಆಗಿಲ್ಲ ಅಂದರೆ they go for chemical method. So chemical method is at least should be considered in integrated pest management. Okay, ಬಟ್ ಎಲ್ಲದನ್ನು ಕಂಬೈನ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಅದಕ್ಕೆ ಅದನ್ನು ಇಂಟಿಗ್ರೇಟೆಡ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಎಲ್ಲದನ್ನು ಯೂಸ್ ಮಾಡಿ ಯಾವುದೇ ಮೆಥಡಲ್ಲೂ ಕೂಡ ಅದನ್ನು ಕಂಟ್ರೋಲ್ ಮಾಡಬಹುದು, ದಟ್ ಈಸ್ ಇಂಟಿಗ್ರೇಟೆಡ್ ಮೆಥಡ್. ಓಕೆ, ಅಂದರೆ ಮೂರನ್ನು ಯೂಸ್ ಮಾಡಿ ಯಾವುದೋ ಒಂದರಲ್ಲಾದರೂ ಕಂಟ್ರೋಲ್ ಆಗುತ್ತೆ, ದಟ್ ಈಸ್ ಇಂಟಿಗ್ರೇಟೆಡ್. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Conversion of sugar into alcohol during fermentation is due to the direct action of temperature. Of course temperature can influence it. Microorganism obviously. So function of microorganism leads to conversion of sugar into alcohol. Then the zymase, concentration of sugar solution. See there, microorganisms would be the one of the option. Okay, microorganism ಇಂದರೆ sugar in the alcohol produce ಆಗುತ್ತೆ. But you cannot choose this microorganism as a right option because ಅದಕ್ಕಿಂತನೂ most precise answer is there, that is called zymase. Okay, so zymase is an enzyme which is present in yeast that converts sugar into alcohol. So ಇಲ್ಲಿ perfect answer zymase ಇರೋದ್ರಿಂದ you cannot choose this microorganism. Okay, so you need to choose zymase. Okay, so artificially the zymase can be isolated from yeast cell itself, that can be used in the production of alcohol. Okay, so remember, most perfect answer ಇದ್ದಾಗ ಅದರ alternative answer ಗೆ you should not consider. Okay, so ಆ ರೀತಿ ಏನಾಯಿತು ಅಂದರೆ ಆಪ್ಷನ್ ಎ ಆ್ಯಂಡ್ ಬಿ ಆ ಥರ ಬಿ ಆ್ಯಂಡ್ ಸಿ ಇದ್ದರೆ ಯು ಕ್ಯಾನ್ ಚೂಸ್ ದಟ್ ಒನ್. ಓಕೆ, ಇದರಲ್ಲಿ ಮೋಸ್ಟ್ ಪ್ರಿಸೈಸ್ ಯು ನೀಡ್ ಟು ಚೂಸ್. ಓಕೆ ಓಕೆ, so which of the following is used in the manufacture of alcohol? As already told, yeast. So yeast is most preferred, that's why it is called as brewer's yeast. Okay, it is called baker's yeast as well as brewer's yeast. So brewer is in the brewing industry, the alcohol producing industry called brewing industry, they prefer yeast. Okay, because it contains a zymase, it can convert and it can produce more alcohol. Okay, next come to question number 12. Yeah, so during sewage treatment, biogas produced include which of the following gases? So primary and secondary sludge are mixed into anaerobic digester. In the anaerobic digester, due to the activity of methanogens, gobar gas is going to be produced. This gas preferably mainly consisting of methane. Along with that methane, it gave some bad smell also. That smell is also due to some chemical, right? And when you burn it, little carbon dioxide is also released. So based on that, so remember, oxygen is not produced in anaerobic reaction. Remember, oxygen is absent in anaerobic digester. Okay, if anaerobic digester oxygen is present, no methane gas is going to be produced, because if the oxygen is present, anaerobic bacteria are unable to function, because anaerobic bacteria do not need oxygen. If oxygen is present, their activity is reduced. Okay, the nitrogen sulfide, methane, sulfur dioxide, no, sulfur dioxide is not present at all. Okay, then hydrogen sulfide, nitrogen and methyl, no, nitrogen is not present within the biogas. So ಈ ಮೂರು ಅಲ್ಲ ಅಂದರೆ ಫೈನಲಿ methane is present, hydrogen sulfide is present which gives the smell, typical smell, then carbon dioxide which is released during when you burn it. Okay, so D is the right option. Okay, next move on to question number 13. So sewage purification is done by, so sewage water can be purified and even you can make it into potable, but it's drinking water. Sewage water can be converted into drinking water as well by different techniques. So ಅದರಲ್ಲಿ maximum sewage purification ಗೆ ಬಳಸುವಂಥದ್ದು ಯಾವುದು? Fertilizers, antibiotics and antiseptic. No, these are microbes. Okay, so what kind of microbes are preferable? So usually they are aerobic bacteria. Aerobic bacteria are going to be used. This aerobic bacteria reduces BOD. Along with that, some fungal mat. So the fungi are also involved. Okay, so this fungi and bacteria together forms flocks. Okay, so when the pumped from aeration tank into sediment tank, part of the bacteria and fungi settles at the bottom, forms the activated sludge. Some part is pumped back into aeration tank, it acts as an inoculum. Okay, ಅದರಲ್ಲಿ ಬರುತ್ತೆ ಈಗ bacteria ಮತ್ತು fungi. So in fungal hyphae is there, within that fungal hyphae bacteria are going to be trapped to form a mat like structure called as a flock. Okay, so aerobic bacteria were used preferably to purify the water. Later the mixture waste material which is produced in primary tank as well as secondary tank, that can be made into mixed into one more tank where anaerobic digester produces the fuel, that is methane. Okay, yeah, so microbes is the right answer. Yeah, first is a direct question. Question number 14. Yeah. A free living nitrogen fixing cyanobacteria which can also form symbiotic association. Okay, so from symbiotic association with the water fern Azolla. So water fern, water iruvanta fern, it belongs to pteridophyte, and Azolla. Azolla is a rootless pteridophyte which is float on the water. So this Azolla increases the fertility of land. Okay, especially rice field. So this Azolla can trap more nitrogen due to the association of this Anabina. So Anabina is a blue-green algae. Okay, so this Anabina helps in conversion of atmospheric nitrogen into ammonia, so it increases the nitrogen content of the soil. Okay, so nitrogen fixing cyanobacteria is Anabina. Okay, so chlorella is a blue-green algae. Okay, even nostoc also helps in nitrogen fixing, okay, but it do not shows any association, so it is a free living organism. But azolazalliruvanಥ associated organism is Anabina. It is a filamentous cyanobacteria, long thread ಥರ ಇರುತ್ತೆ. Okay, it is Anabina. So next question number 15. So which of the following bacteria convert milk into curd? Direct question, there is no doubt about it, it is a lactobacillus. Okay, then Propionibacterium shermani, Swiss cheese, Streptococcus pneumoniae, that is different, and a streptokinase enzyme is going to produce bacillus. Okay, it's a common rod shaped bacteria. So even lactobacillus is also rod shaped bacteria, just like a bacillus. Okay, so bacillus is a term which is used for rod shaped bacteria. Okay, next question number 16. Which role applied by lactic acid bacteria in our stomach? So stomach ಅಲ್ಲಿ ಏನಾದರೂ ಪ್ಲೇ ಮಾಡೋದು lactic acid bacteria, that is labs, of course they are beneficial. Okay, so they form beneficial activity. So beneficial activity ಇರುತ್ತೆ, they can boost up the immune system. Okay, of course the direct question, very simple as well as direct question. Okay, next. The large vessel for growing microbes on an industrial scale are called, okay, big vessel where we can culture the microbes. Big vessel where microbes are cultured. Okay, they are called by two different names. For aerobic organisms and for anaerobic organisms. Here different types ಇದೆ. For aerobic, to get the product, it is called as bioreactor. Okay, anaerobic ಆದರೆ it is called fermenter. Okay, ಈ fermenter ಅಲ್ಲಿ ಆಲ್ಕೋಹಾಲ್ ಮತ್ತು ಆಲ್ಕೋಹಾಲ್ ಯೂಶಲಿ ಆಲ್ಕೋಹಾಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ, whereas in bioreactor some medicines can be extracted by using bacteria. Okay, so ಓದಿದ್ರೆ bioreactor biotechnology chapter ಓದಿದ್ರಲ್ವಾ, that bioreactor. Okay, yeah, so it is fermenters. Okay, so those are called fermenters that can be used to obtain the products like alcohol production ಅಲ್ಲಿ maximum they are going to use those fermenters. Okay, next move on to the question number 18. Okay, so the chemical substance produced by some microbes which can kill or retard the growth of other microbes are called toddy, lactic acid, antibiotics and ethanol. Obviously they are called antibiotics. Okay, so antibiotics are the chemicals that prevent the growth or retard the growth of other microorganisms, called as antibiotic, especially bacteria ge we consider ಮಾಡ್ತೀವಿ. Okay, so first antibiotic was penicillin. Alexander Fleming, okay, they identified that antibiotic property of penicillin. So when you grow the bacteria in a media which contains certain bacteria, you grow the bacteria like this. When the morning he observed some bacterial colonies are going to be dead, so he isolated that particular part. What is the responsible for that? He found that the growing of penicillin. So he isolated that penicillin again and extract the chemical and identify that penicillin shows the resistant against that particular bacteria. So those are called as antibiotics. That was the first antibiotics isolated from microorganisms. That is penicillin G. Okay. So next question. Yeah. The bioactive molecule cyclosporin A. So cyclosporin A, it is a biomolecule isolated from trichoderma. Trichoderma polysporum. Trichoderma polysporum in the fungi. It's a fungi which belongs to Basidiomycete. From this fungi, we can isolate a chemical called a cyclosporin A. So that a cyclosporin A would be used to treat the patients who are undergone organ transplant. Okay, so this cyclosporin A is act as a immunosuppressive agent. Immunosuppressive agent. Okay, so immune ಅಂದರೆ ಹೊಸದಾಗಿ ಆರ್ಗನ್ನ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟ್ ಮಾಡಿದ್ರೆ ಅವ್ರ ಬಾಡಿ ಸೆಲ್ಸ್ ಥಾಟ್ ದಟ್ degrading that newly implanted organ. So to avoid that, the patient need to take the cyclosporine A continuously. So it suppress that immune system with respect to that particular organ. So our body cells do not harm that organ implant. The patient need to take the cyclosporine A continuously throughout the year. Okay. Next, question number 20. Yeah. The symbiotic association between fungi and the roots of higher plant is called. So higher plants na roots. So roots alli some ತರ white patches ಎಲ್ಲ grow ಆಗಿರುತ್ತೆ. So white ರೂಟ್ ಸುತ್ತ ಏನೇನೋ ವೈಟ್ ಕಲರಲ್ಲಿ ಕವರ್ ಆಗಿಬಿಟ್ಟಿರುತ್ತೆ. ಸೊ ದಟ್ ವೈಟ್ ಪಾರ್ಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಫಂಗೈ. ಓಕೆ, ಸೊ ದಿಸ್ ಫಂಗೈ ಈಸ್ ಕಾಲ್ಡ್ ಮೈಕೋರೈಸಾ. This is called mycorrhiza. Myco means fungi, rhiza means root. Root ಮೇಲೆ ಬೆಳ್ಕೊಂಡಿರೋ ಫಂಗೈ ಅಂತೇಳಿ mycorrhiza ಅಂದರೆ. Okay, so it is called mycorrhiza. So there are two kinds of mycorrhiza. Okay, suppose if it is a root, ಆ ರೂಟ್ ಮೇಲ್ಗಡೆ ಮಾತ್ರ ಇರುವಂಥ ಫಂಗೈ, ಇದು ಮೇಲ್ಗಡೆ ಮಾತ್ರ ಹಿಂಗೆ ಕವರ್ ಆಗಿರುತ್ತೆ. Those are one kind of fungi which present on the surface, okay, called as ectomycorrhiza. Ectomycorrhiza. And one more kind is there where if it is a root, that fungi which penetrate inside also, ಒಳಗಡೆ ಎಲ್ಲ ಹೋಗಿ ಬೆಳ್ಕೊಂಡಿರುತ್ತೆ ಹಿಂಗೆ. ದಸ್ ಫಂಗೈ ವಿಚ್ ಪೆನೆಟ್ರೇಟ್ ಇನ್ಸೈಡ್ ಆ್ಯಂಡ್ ಗ್ರೋ ಇನ್ಸೈಡ್ ಆಲ್ಸೋ. ನೋಡಿ, ಇದು ಸರ್ಫೇಸ್ ಮೇಲೆ ಮಾತ್ರ ಇರುತ್ತೆ, ರೂಟ್ನ ಸರ್ಫೇಸ್ ಮೇಲೆ. ದೇ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ಸೈಡ್. ಇನ್ ಸಮ್ ಕೇಸಸ್ ದೇ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ಸೈಡ್ ದ ರೂಟ್ ಆಲ್ಸೋ. ಆ ರೂಟ್ ಸೆಲ್ಸಿನ ಒಳಗಡೆ ಸ್ಪೇಸಲ್ಲೆಲ್ಲ ಹೋಗಿ ಬೆಳ್ಕೊಂಡಿರುತ್ತೆ. ಓಕೆ, ದೋಸ್ ಗ್ರೂಪ್ ಆಫ್ ಫಂಗೈ ಆರ್ ಮೈಕೋರೈಸಾ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಎಂಡೋಮೈಕೋರೈಸಾ. ಎಂಡೋಮೈಕೋರೈಸಾ. Okay, ectomycorrhiza and endomycorrhiza. So what is the function of this ectomycorrhiza and endomycorrhiza? So even we consider plant as autotrophs. They do not depend on any other organisms, we told, but never plant also depends on certain organisms like this fungi, because this fungi which are present in the soil, they are able to give the nutrients, especially phosphorus. Phosphorus na plants ಗೆ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೊಳ್ಳೋಕೆ ಈ ಫಂಗೈ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. Most of the fungi helps the plant to absorb phosphorus. Okay, so fungi provide the nutrients. Nutrient-ಗೆ fungi ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗಿರುತ್ತೆ plants. Okay, yeah, in that strict sense, even plant are also not autotrophs. Okay, so ectomycorrhiza and endomycorrhiza. So one specific example is there for endomycorrhiza. You need to remember that organism name is glomus. Glomus is the endomycorrhiza which gives the nourishment to the plant. Okay, yeah, so those endomycorrhiza and ectomycorrhiza, they are classified under blood group called as a broad group called as a phosphate solubilizing fungi, PSF anta heltare. Phosphate solubilizing fungi that gives the phosphate to plants. Okay, yeah. So this is about mycorrhiza. Remember that organism name, glomus. Okay, so that glomus belongs to a group of fungi who are called as WAM. ಅದನ್ನು ವ್ಯಾಮ್ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ವ್ಯಾಮ್ ಫಂಗೈ ಅಂತೇಳಿ. ಸೊ ಇಟ್ ಇಸ್ ಟೈಪ್ ಆಫ್ ಎಂಡೋಮೈಕೋರೈಸಾ ಓನ್ಲಿ. ವ್ಯಾಮ್ ಅಂದರೆ ವೆಸಿಕ್ಯುಲಾರ್ ಆ್ಯಂಡ್ ಅರ್ಬಸ್ಕ್ಯುಲಾರ್ ಮೈಕೋರೈಸೆ ಅಂತೇಳಿ. ಸೊ ಏನದು ವೆಸಿಕ್ಯುಲಾರ್ ಸ್ಪೆಕ್ಯುಲಾರ್ ಮೈಕೋರೈಸೆ ಅಂದರೆ, ಸೊ ಯಾವಾಗಲೇ ತೊಳೆದಿಲ್ಲ, ರೂಟ್ ಒಳಗಡೆ ಸರ್ಟನ್ ಫಂಗೈ ವಿಚ್ ಆರ್ ಪೆನೆಟ್ರೇಟ್ ಇನ್ಸೈಡ್, ಔಟ್ಸೈಡ್ ಇರುತ್ತೆ, ಇನ್ಸೈಡ್ ಫಂಗಲ್ ಐಫೆ ಇರುತ್ತೆ. ಈ ಐಫೆ ಪ್ರೊಡ್ಯೂಸಸ್ ಅ ವೆಸಿಕಲ್ ಲೈಕ್ ದಿಸ್. ವೆಸಿಕಲ್ ಅಂದರೆ ಈ ಥರ ರೌಂಡಿಗಿರೋ ಸ್ಟ್ರಕ್ಚರು. ಅರ್ಬಸಿಕ್ಯುಲಾರ್ ಅಂದರೆ ಈ ಥರ ನೆಟ್ವರ್ಕ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ನ ರೂಟ್ ಥರ ಸ್ಟ್ರಕ್ಚರ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರುತ್ತೆ. ಸೊ ದೀಸ್ ಆರ್ ಕಾಲ್ಡ್ ವೆಸಿಕಲ್. ಓಕೆ, ನೀವು ಗಾಲ್ಗೆ ಕಾಂಪ್ಲೆಕ್ಸಲ್ಲೆಲ್ಲ ಓದಿದ್ರಿ ಅಲ್ವಾ? ಸೊ ಈ ರೀತಿ ಟಿಪ್ನಲ್ಲಿ ವೆಸಿಕಲ್ ಆಗಿ ಪಿಂಚ್ ಆಫ್ ಆಗಿ ಸಪ್ರೇಟ್ ಆಗುತ್ತೆ. ಅದೇ ಥರ ವೆಸಿಕಲ್ ಫಾರ್ಮ್ ಆಗಿರುತ್ತೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ವೆಸಿಕ್ಯುಲಾರ್. ಈ ಥರ ನೆಟ್ವರ್ಕ್ ಥರ ಸ್ಟ್ರಕ್ಚರ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರೋದನ್ನು ಅರ್ಬಸಿಕ್ಯುಲಾರ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಆ ಥರ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋ ಒಂದು ಗ್ರೂಪ್ ಆಫ್ ಫಂಗೈ ಈಸ್ ಗ್ಲಾಮಸ್, ಕಾಲ್ಡ್ ಆಸ್ ವ್ಯಾಮ್ ಫಂಗೈ. ಸೊ ವ್ಯಾಮ್ ಫಂಗೈ ಆಲ್ಸೋ ಕಾಂಟ್ರಿಬ್ಯೂಟ್ ಟು ಪ್ರೊವೈಡ್ ದ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಟು ದ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, ಸೊ ಇಟ್ ಈಸ್ mycorrhiza. So biofertilizer, we can give one example for biofertilizer, that is glomus. Glomus is one biofertilizer. Okay, and even trichoderma ನಾಟ್ ಸೈಕ್ಲೋಸ್ಪೋರಿನ್ ಏಸೋಲೇಟೆಡ್ ಫಂಗೈ. ಟ್ರೈಕೋಡರ್ಮಾ ಪಾಲಿಸ್ಪೋರಮ್ ಅಲ್ಲ, ಹಾರ್ಸಿಯಾನ ಅಂತ ಇದೆ. Trichoderma harziana. So it is used as a biofertilizer which increases the soil fertility, okay, in case of fungi. Okay, next move on to the question number 31. Okay, the bacterium that commonly lives in animal and human intestine, okay, as a gut fungi. So gut bacteria animals alli matte human alli iruvanta fungi. It is a beneficial fungi. That beneficial fungi is Bacillus anthracis, which cause anthrax. It is not the bacteria. Vibrio cholerae, which causes cholera. No. Then Corynebacterium, which causes food poisoning. So Escherichia coli. Escherichia coli ಅಥವಾ E. coli ಅಂತ ಕಾಮನ್ ಆಗಿ ಕರೆದ್ಬಿಡ್ತೀವಿ. Okay, E. coli. So this is the right answer. Okay. Yeah, next question number 22. Okay. So which of the following serve as a biofertilizer in paddy field? So biofertilizer ಆಗಿ ಪ್ಯಾಡಿ ಫೀಲ್ಡಲ್ಲಿ. ಸೊ ಫಂಗೈನ ಯೂಶಲಿ ಇರೋಕ್ಕಾಗಲ್ಲ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಪ್ಯಾಡಿ ಫೀಲ್ಡಲ್ಲಿ ಹಾಕಿ ಅಂದರೆ ಅಲ್ಲಿ ವಾಟರ್ ನಿಲ್ಲಿಸಿರ್ತಾರೆ. ಸೊ ವಾಟ್ರಲ್ಲಿ ಇರುವಂಥ ಆರ್ಗ್ಯಾನಿಸಮೇ ಆಗಬೇಕು. ದೋಸ್ ವಿಚ್ ಹ್ಯಾವ್ ದ ಎಬಿಲಿಟಿ ಟು ಲೀವ್ ಇನ್ ವಾಟರ್ ದಟ್ ಕ್ಯಾನ್ ಬಿ ಯೂಸ್ಡ್ ಆಸ್ ಎ ಬಯೋಫರ್ಟಿಲೈಸರ್ಸ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಪ್ಯಾಡಿ ಫೀಲ್ಡ್. ಓಕೆ, ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾ ನೋ, ಈಸ್ಟ್ ನೋ, ದೆನ್ ಸೈನೋಬ್ಯಾಕ್ಟೀರಿಯಾ, ಫಂಗೈ ನೋ. Okay, only cyanobacteria can grow there as a biofertilizer. Since blue green algae, let me give an example. For blue green algae, it is nostoc. So this nostoc has a filamentous structure. So it has one specialized cell which resembles H called as heterocyst. This heterocyst, due to the presence of this heterocyst, it has the ability to fix the nitrogen and convert it into ammonia. So nitrogen fixation involve agate, so it increases the soil fertility. Okay, so more nostoc is found abundant in paddy field. Okay, ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಸಮ್ಗಿಂತ ಇದರಲ್ಲಿ ಜಾಸ್ತಿ ಯಾವುದಿರುತ್ತೆ ಪ್ಯಾಡಿ ಫೀಲ್ಡಲ್ಲಿ ಅಂದರೆ it is nostoc. Okay, so nostoc increases the fertility of paddy field. Okay, it belongs to cyanobacteria. Okay, next question number 23. So the solids which are settles after primary treatment of sewage are called, okay, that settles in the first tank. So in the first tank, that sewage water is pumped into one tank where the tank is provided with some filters. Ithra filters irutte where water is made into fall and debris, physical debris are going to be collected. Then water is filled into the tank. Okay, so that water is called as effluent. Okay, so ಇದನ್ನ ಹಾಗೆ ಅಲೋ ಬಿಡ್ತಾರೆ ಸ್ವಲ್ಪ ದಿನ. ಅದೇನಾಗುತ್ತೆ, ಇಲ್ಲಿಂದ ಫಿಲ್ಟ್ರಿಂದ ಪಾಸಾಗಿ ಹೊರಗೆ ಬಂದಿರೋದೆಲ್ಲ ಕೆಳಗಡೆ ಸೆಟ್ಲ್ ಆಗುತ್ತೆ. ಓಕೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ sludge. Okay, and this sludge is produced during primary treatment. So ಇದನ್ನ ಪ್ರೈಮರಿ, ಇಲ್ಲಿಂದ ಬರೋದು ಮುಂದಕ್ಕೆ ಬರೋದೆಲ್ಲ ಸೆಕೆಂಡರಿ ಟ್ರೀಟ್ಮೆಂಟ್. ಸೊ ಪ್ರೈಮರಿ ಟ್ರೀಟ್ಮೆಂಟಲ್ಲಿ ಇಟ್ ಪ್ರೊಡ್ಯೂಸಸ್ ದ sludge, that's why it is called as primary sludge. Okay, so later after the secondary treatment, this primary sludge and secondary sludge are mixed together in a anaerobic digester to produce the gas. Okay, so it is primary sludge. So activated sludge, it consisting of fungi plus aerobic bacteria. Aerobic bacteria irutte idralli, that is called activated sludge. Actually in aeration tank it is floated on the water. Okay, whereas in a sediment tank it settles at the bottom. Okay, activated sludge. So that is called as phlox also. So phlox is a fungal and aerobic bacteria which float on the water, called as a phlox. When it settles at the tank, in a sediment tank, it will become activated sludge. Okay, part of that activated sludge put back into the aeration tank as a inoculum. Okay. Next. So which of the following scientists showed that Saccharomyces cerevisiae causes the fermentation forming products such as beer and buttermilk? Okay, so ಇದನ್ನು ಫಸ್ಟ್ ಫೈಂಡ್ ಔಟ್ ಮಾಡಿದವರು ಯಾರು ಅಂದರೆ, ಸೊ ಡೈರೆಕ್ಟ್ ಕ್ವಶನ್, ಇಟ್ ಈಸ್ ಲೂಯಿಸ್ ಪ್ಯಾಸ್ಚರ್. ಓಕೆ, ದೆನ್ ಅಲೆಕ್ಸಾಂಡರ್ ಫ್ಲೆಮಿಂಗ್, ಪೆನ್ಸಿಲಿನ್. ಓಕೆ, ಸೊ ಲೂಯಿಸ್ ಪ್ಯಾಸ್ಚರ್ ಇಸ್ ಅ ರೈಟ್ ಆನ್ಸರ್. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ನಂಬರ್ 25. Yeah, so germinating barley is employed for the preparation of, this barley is made into ferment in a huge tank under the influence of yeast that going to produce beer. So beer is a product of fermented barley. Okay, so fermented barley in the beer na produce ಮಾಡ್ತಾರೆ. So whereas wine is produced from fermented fruit, okay, grapes, fermented grape ಇಂದ lactic acid milk ಇಂದ produce ಆಗುತ್ತೆ. Cheese by the activity of Propionibacterium shermani and Roquefort cheese, okay, Penicillium roqueforti. By using those we can produce the cheese, different kind of cheese which have different texture, different taste and different aroma. Okay, next question number 26. Okay, so enzymes are used in detergents. So enzyme used in detergent to remove the oil styles. Okay, so oils are fats are nothing but lipids, so lipase is going to be used. So these lipases can be isolated from different organisms like some fungi called aspergillus. Some aspergillus species are even from mucor species. Those lipase can be isolated. Okay, yeah, that's it. So lipase are going to use it in detergent to remove the oil stains. Okay, next question number 27. So primary sludge is used for the preparation of compost, preparation of manure, biogas production, all of this. So primary sludge ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ? So primary sludge is going to be mixed. Okay. So primary sludge is going to be mixed along with the secondary sludge that can be produced biogas plant in a digester. Okay, then after that in a biogas plant digester, they can remove the waste again after the production of gas. That can be dried and used as the manure directly, or it can be mixed with other thing to make it compost. Okay, so primary sludge can be used for all the purpose, manure agi, compost agi, and even biogas production elladukku use ಮಾಡ್ತಾರೆ. Okay, next question number 28. Yeah, so passage of effluent into oxidation tank is for, okay, so ಈ ವಾಟರ್ನಲ್ಲಿ ಎಫ್ಲುಯೆಂಟ್ನ ಹಾಕಿ ಫಿಲ್ಟರ್ ಮಾಡಿ ಉಳಿದಿರೋ ವಾಟರ್ನ when BOD is more, this water is pumped into aeration tank where it is fitted with fans. Okay, ಪ್ರೊಪೆಲೆಂಟ್ ಫಿಕ್ಸ್ ಮಾಡಿರ್ತಾರೆ. ಇಲ್ಲಿ ಏರೇಷನ್ ಟ್ಯಾಂಕ್, ಇದು ಏರೇಷನ್ ಕೊಡಿ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಏರ್ನ ಪ್ರೊವೈಡ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. ದಟ್ ಮೀನ್ಸ್ ಆಕ್ಸಿಜನ್ ಇಸ್ ಪ್ರೊವೈಡೆಡ್ ಟು ರೆಡ್ಯೂಸ್ ದ BOD. ಓಕೆ, ಸೊ ಈ ಪ್ರೋಸೆಸ್ ಏನಿದೆ ಇಲ್ಲಿ ತನಕ ಇದು ಪ್ರೈಮರಿ ಟ್ರೀಟ್ಮೆಂಟ್, ಅಲ್ಲಿಂದ ಬರೋದು ಸೆಕೆಂಡರಿ ಟ್ರೀಟ್ಮೆಂಟ್. ಸೊ ಪ್ಯಾಸೇಜ್ ಆಫ್ ಎಫ್ಲುಯೆಂಟ್ ಇಂಟು ಆಕ್ಸಿಡೇಷನ್ ಟ್ಯಾಂಕ್. So it has two names. One is oxidation tank or aeration tank. Oxidation tank or aeration tank. It's called by two different names. The purpose of filling the water, that is effluent into this oxidation tank, is to reduce BOD. BOD reduce ಮಾಡಲಿಕ್ಕೆ ಅಂತಾನೆ ಈ ಟ್ಯಾಂಕಿಗೆ ಹಾಕ್ತಾರೆ. ಸೊ ಆಫ್ಟರ್ ರಿಡಕ್ಷನ್ ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಪಂಪ್ಡ್ ಇಂಟು ಸೆಡಿಮೆಂಟ್ ಟ್ಯಾಂಕ್. ಅಲ್ಲಿ ವೇಸ್ಟ್ ಎಲ್ಲ ಮತ್ತೆ ಸೆಟ್ಲ್ ಆಗಲಿಕ್ಕೆ ಅದೇ ಪಂಪ್ಡ್ ಇಂಟು ವೇಸ್ಟ್ ಟ್ಯಾಂಕ್. Okay, so passage of effluent into oxidation tank is to for secondary treatment. Okay, so in the primary treatment physical debris are removed. Physical debris removed. Whereas in secondary treatment BOD reduced. BOD reduced. Tertiary treatment, it is nothing but chlorination. ಓಕೆ, ಉಳಿದಿರೋ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾನೆಲ್ಲ ಕಿಲ್ ಮಾಡಲಿಕ್ಕೆ ಟರ್ಷರಿ ಟ್ರೀಟ್ಮೆಂಟ್ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಓಕೆ, ಆಫ್ಟರ್ ದಟ್ ಒನ್ ಮೋರ್ ಇಸ್ ದೇರ್ ಕಾಲ್ಡ್ ಕ್ವಾಟರ್ನರಿ ಟ್ರೀಟ್ಮೆಂಟ್. ಸೊ ಇನ್ ಕ್ವಾಟರ್ನರಿ ಟ್ರೀಟ್ಮೆಂಟ್ RO, ವಾಟರ್ ಇಸ್ ಟ್ರೀಟೆಡ್ ಸಬ್ಜೆಕ್ಟೆಡ್ ಟು RO ಟು ಮೇಕ್ ಇಟ್ ಡ್ರಿಂಕಿಂಗ್ ವಾಟರ್ ಆಸ್ ವೆಲ್. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್ ನಂಬರ್ 29. ಓಕೆ, ಸೊ Monoascus purpureus is a yeast used commercially in the production of ethanol, streptokinase for removing clots from the blood vessel, citric acid, blood cholesterol lowering statins. So Monoascus purpureus, they yield a chemical group called statins. So these statins inhibit cholesterol forming enzyme. Cholesterol produce ಮಾಡೋ enzyme ಜೊತೆ ಕಾಂಪೀಟ್ ಮಾಡುತ್ತೆ. ಇಟ್ ಇನಿಬಿಟ್ ದ ಫಂಕ್ಷನ್, ಸೊ ಇಟ್ ರೆಡ್ಯೂಸಸ್ ದ ಬ್ಲಡ್ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಲೆವೆಲ್. ಸೊ ಬ್ಲಡ್ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಲೆವೆಲ್ನ ಕಡಿಮೆ ಮಾಡುತ್ತೆ. ಓಕೆ, ಸೊ ದಟ್ ಇಸ್ Monoascus purpureus. ರಿಮೆಂಬರ್ ದ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ ನೇಮ್ Monoascus purpureus that yields statins. So similarly streptokinase produced from Streptococcus bacteria, it used as a clot buster to remove the clots in blood vessels. Okay, next question number 30. The same question, sorry, the repeat ಆಯ್ತು ಮತ್ತೆ. Statins are obtained from Monoascus purpureus. Okay, so this streptococcus, streptokinase Mucor javanicus lipase enzyme, Clostridium botulinum, it yields food poisoning, it causes food poisoning. Then butyric acid also, butyric acid is produced from the Clostridium butyricum, butyric acid is produced. Okay, so it is Manascus purpureus. 30, next 31. So organic acid firstly produced through the fermentation is, fermentation ಮಾಡಿದೆ ಫಸ್ಟ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುವಂಥ ಆರ್ಗ್ಯಾನಿಕ್ ಆಸಿಡ್ ಯಾವುದು? ಆರ್ಗ್ಯಾನಿಕ್ ಆಸಿಡ್ ವಿಚ್ ಪ್ರೊಡ್ಯೂಸ್ ಡ್ಯೂರಿಂಗ್ ಥ್ರೂ ಫರ್ಮೆಂಟೇಷನ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್. So it is lactic acid. Okay, so just you recall the respiration chapter in the first year, where glucose is converted into pyruvic acid. This pyruvic acid, either it can convert it into ethanol or lactic acid. Lactic acid dehydrogenase enzyme. Okay, lactic acid dehydrogenase. Just remember the name. Lactic acid dehydrogenase. In the presence of lactic acid dehydrogenase, pyruvic acid converted into lactic acid. Whereas ethanol, so ಇಲ್ಲಿ ಎರಡು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಥವಾ ಎಂಜೈಮ್ ಬೇಕಾಗುತ್ತೆ. ಒಂದು ಕನ್ವರ್ಷನ್ ಆಫ್ ಅಸಿಟಾಲ್ಡಿಹೈಡ್ plus a CO2. So it is by pyruvate decarboxylase. Then acetaldehyde converted into ethanol. It is ethyl alcohol dehydrogenase. Ethyl alcohol dehydrogenase is an enzyme where it converted acetaldehyde into ethanol. Okay. Yeah, so this is how the lactic acid is produced in muscle cells in the deprived of oxygen. Oxygen kadme adaga ee lactic acid start to produce. Okay, so this citric acid is a product of Krebs cycle. Okay, it's the first compound produced during citric acid. That's why it is called as a citric acid cycle. Okay, then oxalic acid. So oxalic acid is present in some plant called Oxalis corniculata. Okay, next question number 32. So in cheese, microorganisms are required for. So cheese na ripen madistare by using microorganisms. Why the cheese is inoculated with the microorganisms andre, so it gives sour taste as well as ripening. Souring ಮತ್ತೆ ripening, specific taste na produce ಮಾಡುತ್ತೆ ಮತ್ತೆ ripening ಆಗುತ್ತೆ. For that purpose so microorganisms are inoculated to cheese, like a Penicillium roqueforti, which gives a specific color and a specific taste and specific aroma to that cheese. That's why they are going to use that microorganisms. Example for this souring and ripening for cheese, you can give the best example. One is Penicillium roqueforti, another one is Propionibacterium shermani. Okay, next 33. So glucose fermentation of yeast yields, so no doubt it is ethanol plus carbon dioxide. Okay, so after pyruvic acid, so pyruvic acid to pyruvate dehydrogenase, it produces acetaldehyde, so carboxylase. Then acetaldehyde is converted to ethanol by the activity of alcohol dehydrogenase enzyme. Okay, next 33, 34. So microbes use it for the biocontrol of a pest, butterfly caterpillars. So butterfly caterpillars can be controlled by using, as already told, it is controlled by using Bacillus thuringiensis spores. When this Bacillus thuringiensis spores are going to be sprayed on the plant, those spores, when this ingested by this butterfly caterpillars, so they are going to die because this Bt toxin made the spores in the gut of that insect that going to kill. Okay, so trichoderma can be used as fungi that can prevent the growth of other fungi. Okay, so adanna bere fungi na root borne pathogen control madlikke we can use the trichoderma as a biocontrol agent. Saccharomyces cerevisiae, it is a yeast. Then a streptococcus species, streptokinase can be isolated. Okay, and even some streptococcus pneumoniae is also there. Sorry, staphylococcus pneumoniae. Okay, then 35. So lactic acid bacteria convert milk into curd and improves its nutritional quality by enhancing which vitamin? So it can increase vitamin B. Okay, vitamin B. So when the lactic acid bacteria act on the milk protein, it breaks down to milk protein and make into clumpy. That leads to the formation of curd. Okay, so which rich with vitamin B. Okay, next question number 36. So chloramphenicol and erythromycin, they are the broad spectrum of antibiotics, are produced by Streptomyces, Nitrobacter, Rhizobium and Penicillium. So Streptomyces, remember actinomycetes. So Streptomyces belongs to group called actinomycetes. Actinomyces. So these are the bacteria, but they have fungal property. That's why they are called as actinomycetes. Okay, actinomycetes. Myco means fungi. They resemble the property of fungi also. They produce hyphae-like structure. That's why they are called Streptomyces. So whatever the antibiotics we are going to use presently, most of those antibiotics are isolated from this Streptomyces species. So these Streptomyces have the ability to kill the bacteria. Okay. So for example, chloramphenicol is one and erythromycin is one of the antibiotic which is isolated from Streptomyces species. Okay. So Nitrobacter, nitrogen fixation time then baratta, then Penicillium, penicillin G, then Rhizobium, nitrogen fixation. Okay. Yeah. So here next question, methane content of biogas. It is direct question, 65%. So along with that, it also has hydrogen sulfide as well as carbon dioxide. Okay, so methane has 65% of methane in it. Okay, next question number 38. Okay, so baculoviruses, a nucleopolyhedrovirus, they are called as NPHV, nucleopolyhedrovirus, does not show. Okay. So host specificity, narrow spectrum applications, effect on non-target pathogens, utility in IPM program. Of course, that can be used in IPM program, and they do not show effect on non-target pathogens. They can kill only a specific group of insects. Okay, that's why they are called as very specific. They do not target other organisms, only kill pathogens. So called as a nucleopolyhedrovirus. NPHV can be used as a biocontrol agent. So it's a virus. Nucleopolyhedrovirus can be used to control insect pests. Okay. Next question number 39. So primary treatment of sewage is a physical process, biological process, chemical process and biochemical process. So it is a physical process. Primary treatment, we are going to remove the physical debris. So it is a physical process. So biological process or chemical and biochemical process, they comes under secondary treatment. Especially biological process, the biochemical oxygen demand, it is a secondary process. This is also secondary process. Chemical process, tertiary. Treatment with the chlorine is a chemical process. So primary treatment is physical process to remove the debris. Okay. So whereas biological and biochemical both are comes under secondary in terms of decrease in BOD. So whereas chemical treatment for chlorination. Okay. So this is about the thing. So primary treatment of sewage is obviously it is a physical process. Okay. So next question. Sewage treatment process in which a part of a decomposed bacteria is recycled into starting of the process is called as. Okay. So sewage treatment ali secondary aeration tank in the when the BOD is reduced. So ಯಾವಾಗ BOD reduce ಆಗುತ್ತೋ ಇದ್ರ ಮೇಲೆ ಫ್ಲಾಕ್ ತೇಲ್ತಾ ಇರುತ್ತೆ. Along with the flock, it is pumped into one more tank and allow for settle. ಇಲ್ಲಿರೋದೆಲ್ಲ ಕೆಳಗಡೆ ಹೋಗಿ ಸೆಟ್ಲ್ ಆಗಿಬಿಡುತ್ತೆ. Okay, now water purify ಆಗ್ತಾ ಇರುತ್ತೆ. Some part of this sludge ಅಂದರೆ ಕೆಳಗಡೆ ಉಳ್ಕೊಂಡಿರುವಂಥದ್ದು, some part of this sludge put back into the aeration tank as a source of inoculum. Okay, so ಯಾಕೆ ಅದನ್ನು ಆ್ಯಡ್ ಮಾಡ್ತೀವಿ as a inoculum ಅಂದರೆ, so ಈ sewage water the amount of aerobic bacteria is very less. To reduce the BOD in this tank, they took many days. If you add a source, ಇಲ್ಲಿಂದ ಇನ್ನೊಂದಿಷ್ಟು aerobic bacteria ಆ್ಯಡ್ ಮಾಡಿದ್ರೆ the process of BOD reduction increases. ಬೇಗ ಆಗಿಬಿಡುತ್ತೆ, ಒಂದು ವಾರದಲ್ಲಿ ಆಗೋದು 3 ಡೇಸಿಗೆ ಆಗುತ್ತೆ. That is why we are going to use as an inoculum. Okay, that is called activated sludge. Okay, so activated sludge treatment is called activated sludge treatment. So due to the activated sludge treatment, it increases the rate of reaction with respect to reduce the organic matter in the water. That is BOD ಬೇಗ ಕಡಿಮೆ ಆಗುತ್ತೆ ವಾಟರಲ್ಲಿ. Okay, so that's it. This is all about the chapter microbes in human welfare. Thank you.